Hello, and welcome to the Arthritis National Research Foundation's Research Spotlight webinar series. I'm Kaylin Young, the Director of Corporate Affairs with ANRF. And on behalf of our Board of Directors, our Scientific Advisory Board, and our staff, I would like to welcome you to our Spotlight on Psoriatic Arthritis. We're so excited to have so many of you joining us today. We would also like to recognize and thank each of our corporate partners for supporting our Research Spotlight series. Thank you to AbbVie for being a gold level sponsor, for your continued partnership and dedication. It's because of our partnerships that we're able to continue to pave the way for innovation with our researchers. So thank you. Before we get started, we wanted to go over just a few logistical pieces of information. The first is this webinar is being recorded and will be available on our website and our YouTube channel in the coming weeks. The second is, if you have any questions, please type them into the chat, the Q&A chat box. And if we're unable to get to your question during the webinar, you can always email your questions to info@curearthritis.org after the webinar. I'm now honored to turn the mic over to our patient moderator, Lacey, who will share her story and get us started. Thank you so much, Lacey. It's all yours.
Thanks so much, Kaylin. Hello, my name's Lacey. I was 20 years old when I first started having joint pain. Waking up in the morning would take longer than I was used to because I had such a hard time getting out of bed. My lower back was stiff and painful, and I didn't understand what was going on with my body. I worked at the front desk of the Washington University Rheumatology Clinic, but I didn't want to abuse my position by asking one of my physician friends what they thought. I didn't have a choice after I woke up in the middle of the night with my knee swollen and painful to the point I was unable to walk. I sent a text with a photo of my scary looking knee to my friend, Dr. Richard Brasington, to see what he thought. He saw me in clinic 2 days later when I was finally able to walk. After months of blood work, x-rays, and an MRI, I finally had an answer. I had a rheumatic disease just like the patients I saw coming to clinic every day. It took several years and 5 different medications before we finally found a combination that works for me. There was pain, confusion, frustration, and anxiety from the start of my symptoms to knowing and having a treatment. And that's with me working for rheumatologists. 16 years later, and still working for rheumatologists, I now understand how difficult it is to pin down a diagnosis for rheumatology patients and also the stress and anxiety from a patient's perspective. I'm so thankful that I was in my unique position to be treated as quickly as I was, and I don't take that for granted. I, like you, am looking forward to hearing from our speakers today about their latest research and how they're moving the needle forward to earlier diagnosis. I'd like to introduce our first speaker, Dr. Barhram Razani. He earned his degree in biochemistry from Rice University in Houston, Texas. He then completed both his medical degree and his PhD in immunology at the University of California, Los Angeles. During his PhD, he studied how certain proteins in the body help control immune system responses. After that, he trained in dermatology at the University of California, San Francisco. Since 2017, he has been a part of UCSF's dermatology department and received a grant from ANRF to support his research in 2022. Dr. Razani's work focuses on understanding how skin cells affect the immune system, both in the skin and throughout the body. He is particularly interested in studying psoriasis and its related health problems using mouse models.
Thank you so much, Lacey, for that introduction. As Lacey mentioned, I'm a dermatologist out here in San Francisco, and I have a particular interest in understanding the connection between psoriatic skin and joint disease. Let me move forward here. As a dermatologist, psoriasis is one of the most common inflammatory skin diseases. It affects about 3% of the US population. And interestingly, of that group of individuals with psoriasis, about 30% will go on to develop psoriatic arthritis, which can be quite debilitating, requires often systemic medicines for treatment. This is on the right, we see a particular pattern of psoriatic arthritis that affects primarily the fingers and toes. but one of the fascinating connections between psoriatic skin and joint disease is really this lag period that is typically, although not exclusively, seen in many individuals, where psoriatic skin disease presents first, and then after a period of 5 to 7 years, psoriatic arthritis will present. And this delay has led to the hypothesis that potentially psoriatic skin inflammation in the skin, can either trigger or drive subsequent arthritis. And understanding the connection between these two is not only the focus of my research, but a general interest in psoriatic research. This particular delay is quite important to understand, because we know that delayed diagnosis of psoriatic arthritis can lead to overall poor outcomes for patients. And so capturing those individuals that are going to evolve into a psoriatic arthritis picture early is really quite important clinically, and something that's a focus of the field. So the overarching goals of my research are to help find biomarkers to identify individuals with psoriasis that are at risk of developing psoriatic arthritis, and understand really the immunological mechanisms, the inflammatory mechanisms that connect psoriatic skin and joint disease, to help develop new treatments to prevent this transition. The way that we have begun to understand these immune mechanisms is to really look at the genetics of both psoriasis and psoriatic arthritis. And there have been a number of really beautiful studies looking at the entire human genome and understanding, are there particular variants of genes that are more associated with psoriasis and psoriatic arthritis. And these studies have identified a handful of different genes that particular variants of those increase your risk. And the gene that we've been very focused on is known as TNF-AIP3 or A20. Sorry, I don't know if you can really see my cursor, but this red dot should be over TNF-AIP3. It turns out that particular variants tend to reduce expression of this gene, and individuals with this variant tend to have higher risk of both psoriasis and psoriatic arthritis. Independent of particular genetics, if you look at the skin of patients with psoriasis, levels of A20 are reduced in the skin. And both of these pieces of data together really suggest that this gene A20, which I'll primarily be referring to it as A20, is an important protector against psoriatic inflammation. And so we've been very interested in understanding how this gene functions to prevent both psoriasis and psoriatic arthritis. Again, this gene is really broadly expressed in every cell type. And so one of the questions has been, does A20 function within skin to prevent both psoriatic skin inflammation and psoriatic joint disease? And the particular cell type that we've been very interested in is the keratinocyte. This is really the key cell type of the skin. It's the cell that you really see when you look at your body. So if I'm looking at my hand, I'm really seeing the keratinocytes. It's those cells that make the topmost layer of the skin. And so given this interesting connection between psoriatic skin and joint disease that we see in individuals, we were curious to understand if A20 functions within keratinocytes to prevent psoriatic skin and joint disease. To answer this question, we really have to use mouse models, because we need to actually be able to change the level of A20 in keratinocytes and understand what happens to tissues subsequently. And so we've used this mouse model, which we've termed the A20 EKO mouse. And in this mouse model, on the left, the mice are born completely normal. They have normal levels of A20, but we've used genetic tools to allow for deletion or elimination of A20 just in keratinocytes after the mice's treatment with a particular medicine. And so this is a particularly powerful model because, number one, we know exactly when the inflammatory process begins. Even at the earliest stages, something that we really can't understand or really detect in humans with psoriasis and psoriatic arthritis, because we don't know when the inflammation really started. But in these mice, we know it exactly starts when we treat the mice with medicines. And number two, we know exactly where the inflammation is beginning. It's beginning in the keratinocytes or in the skin. So these mice the photos of them are on the right. These are A20 EKO mice on the right. On the left side, you see A20, what we call E wild type, or the normal mice. These are mice that have no changes in A20 in their keratinocytes. But on the right, you can see that they develop scaliness of the ears and tails. And under the microscope, when we look at the skin, the changes in the skin cells and inflammation really mimic what we see under human psoriasis. We see thickening of the skin, inflammation of the skin. But surprisingly, the changes in these mice don't affect just the skin, even though that's where the change of the A20 is, but also they develop a picture that looks very similar to certain patterns of psoriatic arthritis. So if we looked on the left, the A20E wild type mice, these are normal mice, no changes in A20. You can see that they have these long, thin digits, these long nails. What you're seeing there is histology of a cut through the finger. You can see the nail, the joints. But on the right, on the A20 EKO mice, you can see that these mice have swelling of the digits, progressive loss and destruction of the nail. And on histology, there is destruction of the bones in the tips of the digits, as well as arthritis in the joints and inflammation of the tendons, all features of psoriatic arthritis. And what's interesting is that this inflammation happens in all of the mice over the course of several weeks. So again, the real changes in the skin cells, but we're beginning to see changes or inflammation in the joints subsequently. So there are many ways to use this mice, but one of the ways that we've been very interested in is understanding what are the earliest molecular changes in skin that ultimately lead to the arthritis. And so the way we've tackled that is we've treated the mice with a medicine to delete A20 in the keratinocytes or the skin cells. And only 1 week later, at a time point where we really don't see inflammatory changes yet, everything is really on a molecular level, we did a technique called RNA-seq that evaluates the changes to the levels of all genes across the genome. So there's about 30,000 genes in the genome, and we can detect which ones go up and which ones go down. Now, a lot go up and down, but there are tools to really categorize these genes into different pathways. And two of the pathways that we were really interested in were the defense response to virus, or an antiviral response that seems to happen once we delete A20 in keratinocytes, and also the response to interleukin-1. And interleukin-1 is an inflammatory factor. There's a family of them, and there were genes that were increased that were related to all of them. Now, we use genetic tools to really dissect which of these pathways is the most important in causing the subsequent arthritis. And I'll focus on one, the response to interleukin-1, which had a very interesting result. This pathway is really driven by a protein known as MyD88. And so MyD88 is a critical protein that allows for this response to interleukin-1 pathway to happen. And so our question was, if we remove MyD88, then can we prevent the psoriatic skin and joint disease that happens in these A20 EKO mice? And so you can see that the genetics is on the left. These are the A20 EKO mice, so the mice that we delete A20 in keratinocytes, but simultaneously we've used the genetic tool to delete MyD88 also in the keratinocytes at the same time. So we remove the A20, remove the MyD88 at the same time in keratinocytes, and these mice don't develop any psoriatic skin and joint disease. And so based on this data, we really think that when A20 is eliminated in keratinocytes, then this response to interleukin-1, MyD88 pathway really shoots up and causes all the subsequent inflammation. Now, this is a mouse model, and so we always are interested in understanding whether similar things are happening in human cells. And so we took advantage of the fact that you can culture human keratinocytes in a dish. And so we obtained human keratinocytes from volunteers. These are healthy individuals without psoriasis and psoriatic arthritis. And so on the left, you see the cartoon here. We used CRISPR techniques, and which many of you may have heard of. These techniques allow us to actually delete particular genes in cells. And so we took these cells and we split them into three groups. One of them we just mock treated, so they're sort of the control group. In the middle group, we deleted just A20 in the keratinocytes. And then in the third group, we deleted A20 and MyD88. So we sort of did an experiment like we did in the mice, but in human keratinocytes in a dish. And you can see on the right-hand graph, we're looking at levels of an important inflammatory gene CCL20, which among many different genes that we tested that are related to psoriasis really bring inflammatory genes into tissues. CCL20 is one example of that. You can see that in the control group of keratinocytes, they have very low levels of this gene, but that when you delete A20, CCL20 levels really go up. And if you delete A20 in combination with MyD88, they stay low. And so we really think that when you remove A20 from keratinocytes, it causes these cells to start pumping out inflammatory mediators that can subsequently lead to psoriatic skin and joint disease. And so as sort of a basic model where we are now after this work, we think that in individuals without psoriasis or psoriatic arthritis, there's plenty of A20 in keratinocytes to prevent this type of inflammation, but that when you remove A20, then that allows MyD88 activity to really go up spontaneously, and that this MyD88 activity is really critical for not only causing the psoriatic inflammation, but also ultimately the psoriatic arthritis. And we're really interested in what's going on in this blue arrow. We know that immune cells like T cells are really important in doing this connection. And so that's the goal of subsequent research. I wanted to thank Ryan Tobias in my lab for doing much of the work, collaborators, and really a special thank you to the ANRF, whose generous funding has really been critical for pushing this research forward, and really has been the primary funder of the research that I presented today. So thank you very much to the ANRF and everyone who contributes to that organization.
Thank you so much, Dr. Razani. Don't forget to ask your questions in the Q&A box below. I would like to present our next speaker, Dr. Michael Paley. Dr. Michael Paley is an assistant professor of medicine and rheumatology at Washington University in St. Louis. He earned his bachelor degree from Yale and his MD PhD from the University of Pennsylvania, where his research on T cells was published in Science. After moving to St. Louis, he completed his medical training and a rheumatology fellowship. Dr. Paley now directs the Ocular Rheumatology Clinic and leads a lab studying autoimmune diseases, especially axial spondyloarthritis and uveitis, which is an eye inflammation. In 2022, he helped discover key immune cells involved in these diseases with results published in Nature. An ANRF grant in 2021 supported his work to develop tools that help doctors detect autoimmune diseases early. He believes we are on the verge of a major breakthrough in understanding autoimmune diseases. Take it away, Dr. Paley.
Thanks for that very generous introduction, and thank you for giving me the opportunity to share the work that we're doing in the lab. So today I'm going to be talking about HLA-B27 and hopefully convince you that the work that we're doing in the lab have implications for the connection between psoriasis and psoriatic arthritis, as you just heard about in Dr. Razani's talk. All right, so I'm going to take a little bit of a step back because we were just focused on the connection between the skin and the joint. And I'm going to just talk a little bit more globally about this umbrella or this family of diseases that we call spondyloarthritis, where you have different patients having different organ involvement, but it's relatively idiosyncratic, and we currently lump them all together. So you can get psoriasis in your skin, you can get uveitis, which is inflammation in your eye, you can get sacroiliitis or spondylitis, which is inflammation in your spine and your pelvis, or you can get inflammation in your peripheral joints like your knees and your elbows and your wrists and so on. You can also get inflammation in what we call the entheses, which is the connection between the tendon and the bone. So commonly you'll have the Achilles tendon, the insertion point right into the heel gets inflamed, or you can get inflammation in your gut in the form of inflammatory bowel disease. So like I mentioned, patients can their diseases pick and choose sort of what manifestations they get under this large umbrella. And so one question that I think researchers have been trying to figure out is how are all these different organs connected? So one way that they're connected is it's thought that there's evidence that inflammation in the gut increases your risk of developing a variety of these different other manifestations, right? Which kind of connects inflammatory bowel disease with many of these other manifestations, right? So you can get psoriasis in your skin, spondylitis, or uveitis. And I'll come back to that point a little bit later. The other important risk factor in the genetics that we talked about with Dr. Razani are the HLA genes. So I don't know if you remember from that slide when he was talking about genetic risk factors for psoriasis, the greatest genetic risk factor was in the HLA-C gene, which is refers to the allele or the variant of the HLA-C gene, HLA-C06. And in contrast, in spondylitis and uveitis, there's a similar HLA gene, but this one is HLA-B27, which dramatically increases your risk for developing spondyloarthritis or uveitis. And so my proposal for you today is that the work that we're doing to better understand how HLA-B27 causes spondyloarthritis and uveitis will help us develop a framework to better understand how HLA-C06 may lead to psoriasis and therefore psoriatic arthritis. All right, so let's talk about what these HLA genes actually do. And I'll try to convince you of my proposal. So here I have a cartoon that I stole from a paper about 8 years ago that sort of diagrams or simplifies what's going on. So on the bottom here, you have the cell that's in blue, and all your cells in your body have thousands of different proteins. Here it's diagrammed as a chicken. And then at the end of the lifespan of a protein, they usually end up going to a complex, what's called the proteasome, which here is diagnosed with the magician with the saw. And the purpose of the proteasome is to cut up all these proteins into tiny little pieces called peptides. And what HLA molecules do is they take those pieces and they put them on the surface of the cell. And so the whole point of these HLA molecules is to show off to the immune system what's going on the inside of the cell. And so you have 3 different HLA genes, HLA A, B, and C. You get 1 copy from each parent. And they all present different pieces or different peptides on the surface of the cell to the immune system. And the cell that surveys these HLA molecules is called the CD8 T cell, right? So you can think of this as the cop on the beat, making sure that all these cells are healthy and normal and that there are no issues, right? Because sometimes cells get infected with a virus like influenza or SARS-CoV-2, which causes COVID-19. And so you get basically turns the cell into a viral production factory, and it generates lots of viral proteins. Some of those proteins end up passing through the proteasome into the little viral peptides. And the HLA molecules take those viral peptides and put them on the surface. And then the immune system sees that, oh no, this cell is infected with a virus. We got to get rid of it and target it for elimination, because that's how you control a viral infection, is to eliminate all the infected cells. All right. So here I'm kind of, how this actually works is a little bit more complicated than that cartoon, of course, in that everybody has billions of different T cells, and they all are unique for something specific out there in the universe of viruses and bacteria, right? Because you can encounter, billions of different viruses and bacteria. So here we're showing you 3 different cells. All of them have HLA-B27 on their surface, and they're all presenting a different peptide. And then of all your different T cells, you have a specific T cell for each individual peptide, right? So there's this dark blue one that recognizes this square peptide, and you have the light blue one that recognizes the circle, and the black one recognizes the triangle, right? And so in theory, there's sort of this one-to-one matchup for like a T cell with its own unique T cell receptor specific to a specific peptide. The thing that's been hypothesized for a while and has begun to emerge is that sometimes peptides are actually quite similar to one another, and they actually mimic each other at the molecular level, and they're so structurally similar that the T cell with its unique T cell receptor can't tell them apart, right? And here that's diagrammed on the right. And this is because they're so similar to one another, this is what we call molecular mimicry. And this is a concept that's going to come up in about 2 slides. All right. So the question for us is that the hypothesis for a very long time, since HLA-B27 has been associated with uveitis and spondylitis, is that because the HLA-B27 presents peptides to the immune system, and HLA-B27 is unique in increasing your risk of uveitis and spondylitis, maybe it's unique also in a way that it's presenting a unique peptide or set of peptides to a unique T cell or set of T cells. And so we wanted to ask, well, are there specific T cells in the eye or the joint that are driving disease? So we were really fortunate to try to answer this question in a large multinational or international collaboration across lots of different specialties, where we sampled patients with B27 uveitis or B27 spondylitis. And we took the cells out of the eye and the blood in patients with uveitis and the joint and the blood out of patients with spondylitis. We subjected those cells to this newer technology at the time called single-cell T-cell receptor sequencing, which basically is a fancy way of saying that we can identify every single T cell or every single COP in those inflamed tissues. And what we found is that the very similar or the exact same T cell receptor were in these different patients in these different tissues. And I should mention that these patients were from different continents. But they were enriched in the eye over the blood in the uveitis patients, or in the joint in the blood over the blood in the spondylitis patients by about 10 to 100 fold, which really suggested that there was a shared process, a shared peptide activating a shared T cell. And so we characterized very carefully the T cell receptor that's unique for that specific T cell. And the only thing I would highlight is that it's comprised of TRBV9 that makes up a component of that T cell receptor. And I will come back to that in a second. So you might be wondering, well, what do these T cells actually recognize in the patients? And so we were very lucky, again, to collaborate with the Garcia lab at Stanford, who had developed a platform to sort of identify the peptides that these T cells recognize. And one of the peptides that they found is actually that these T cells recognize a peptide from bacteria. And particularly, this is bacteria that is in your intestines. And so this kind of makes a connection between what's going on in the GI tract and what's going on in the eye and the joint. But the other thing that we found out is that actually these cells also recognize peptides from humans, right? They're in our own genome, in our own cells. And the peptides are so similar to one another that the T cells can't tell them apart, right? And so again, this is the concept of molecular mimicry. And so what we think is happening is that the T cells initially recognize this peptide from bacteria, and they have an appropriate normal immune response to prevent this individual from getting sick, right, and fending off from the bacteria. But unfortunately, those cells then start to recognize pieces of the person's own body, and then they start to think, let's attack that body because there must be something wrong because they think they're seeing the same thing. All right. So to put this all together in a cartoon, the way we think about it is that there's some inflammatory environment or event that happens in the GI tract where the T cells get exposed to this pieces of the bacteria. And then at some point they start entering the general circulation and they end up landing in either the eye or the pelvis. And they end up seeing pieces that look like the pieces of the bacteria in the eye or the pelvis. And therefore they're like, oh, I need to fight off this infection. It's in a different organ, but I'm still seeing the same thing, so it must be eliminated. And that, I think, that confusion, that molecular mimicry, we hypothesize is that the underlying cause of why people are getting inflammation in the eye and in the pelvis. All right. So this is exciting in the sense that I can explain to patients potentially how they got this disease, which is gratifying for some people. But some people really want to know, like, okay, so you got all this knowledge, what are you going to do to help me, right? And so one thing you can think about is like, well, if these are the cells that are causing disease, what if we just get rid of them? Would that work? So a company that's based out of Russia and Belarus had this idea and developed a drug to actually try to do that specific thing. So they developed a drug that gets rid of T cells that use TRBV9 as part of their unique T cell receptor. And I mentioned before that the T cells that are causing disease that we described are all using TRBV9. And so what this drug does is basically gets rid of all the TRBV9 T cells. That's about 5% of all your T cells. So it's going to get rid of all your autoimmune clones. Those are the red triangles on the cartoon there, and some healthy ones too. But because it's only getting rid of 5%, that means that like 95% are totally intact. And that is what we think is enough to protect you against all the viruses and the bacteria out there in the world because of enough redundancy in the immune system. And so the promise of this therapy is that you can specifically target the cells that are causing disease without the general immunosuppression that you would get with, say, a TNF inhibitor like adalimumab or etanercept or an IL-17 inhibitor, or many of the other molecules that we use to treat patients with psoriasis or psoriatic arthritis. So they published the first patient at what seems like that they tried this drug on last year, and I just wanted to go over the case. So this was a patient that was initially diagnosed in 1983, had symptoms in 1983, and then first got diagnosed in 1986. So about 3 years delay, which is, pretty standard, unfortunately, in this disease, and got treated with, NSAIDs like ibuprofen or naproxen and so on. Around the turn of the century, got tested for HLA-B27, which was positive, and then around 2004, got started on, anti-TNF therapy like etanercept or adalimumab. And that was on for about 5 years, and then underwent an experimental stem cell transplant, like a bone marrow stem cell transplant, to figure out if you could reset the immune system and maybe get rid of this disease. And that worked temporarily for about 2 years before the person had the disease come back and then had to be started on therapy again. Didn't do so great, had to have a left hip replacement, but then in around 2019 got started on this new therapy that specifically targets the disease-causing T cells, and actually did so well that the person could come off their anti-TNF therapy and was just on maintenance therapy with this depleting agent for about 4 years. And the bottom row is sort of evidence of that. So these are 3 different graphs that show the disease activity of this person. There's just 3 different measurements. And so you can see on the left, in B, it's like kind of high. And then when you get to the dashed blue line, which is the bone marrow transplant, it goes down. But then, of course, it comes back up. But then when you hit the green dashed area, when he's getting this new therapy, you can see that the disease activity is going down again. And that's kind of true for all 3 graphs. So what I want to highlight is that this is very exciting. It also is done on a patient who, with the exception of the stem cell transplant, which is kind of unique, somebody who'd had disease for decades, right? So this is we're not capturing him just at the onset of disease. Excuse me. We're catching somebody who's had established disease and they're actually getting a real benefit from this therapy and able to come off their other drugs. And so what I hope is that the future of rheumatology is going to follow this example, where we're going to understand the fundamental mechanisms and molecular biology of these diseases so we can target patients and their diseases more sniper-like than the carpet bombing of today, and really get rid of the bad causing cells and leave the rest of the immune system alone so that people can go on and live normal, healthy lives. So with that, I got to thank everybody in my lab who helped me with this work, my collaborators in ophthalmology and Stanford and Oxford and internationally, the patients, as well as the support and funding from the Arthritis National Research Foundation. So with that, thank you very much.
Thank you so much, Dr. Paley. As a reminder, everyone, please ask any questions that you may have in the Q&A box below. We do have one question to start off with. The person asked, oh gosh, hold on, I just hid. I want to make sure that I'm taking something that will treat both the psoriasis and PSA. What is the best course of action for patients who are starting this journey? Oh, Dr. Paley, you're muted.
I was deferring to the dermatologist here because I like to lean on the expertise of my colleagues. But there's plenty of drugs that will treat both. And I think it's always just helpful to have a conversation between the rheumatologist and the dermatologist to see what's going to be optimal. But a number of drugs, the easiest ones to think about for a rheumatologist are like your biologics, like your TNF inhibitors, your IL-17 inhibitors, your IL-23 inhibitors, your JAK inhibitors are all going to be able to treat both your arthritis and your skin disease to varying degrees. And I think it's just a question for each individual patient, what's the best combination for them. But Dr. Razani, I'm sure, has much better ideas.
No, I think that was well said. It turns out that most of the immunosuppressants that work particularly well for psoriasis will work for psoriatic arthritis. I think the question in the field that is interesting is that the therapeutics tend to work much better for psoriatic skin disease than they work for psoriatic arthritis. Even though they do work for psoriatic arthritis, the control is a little bit not as effective. I think that's the question in the field, why that's the case. But in terms of what we have available today, compared to the treatments of the future, really most of the IL-17, TNF, IL-23 inhibitors will really help for both, as well as the JAK inhibitors.
Thank you. The second question we have is, how do you see treatments changing or improving in the next 5 years? It's a big question.
I mean, this person's drinking my Kool-Aid, right? Like, I think we're, in terms of the T cells, right? I'm a T cell guy, so this is what I'm going to talk about. I think we're going to be able to better identify the cells that are causing disease and develop therapies to block them or delete them or what have you. I think there's lots of strategies that are going to be emerging, and I think this is going to be the forefront of autoimmune disease over the next 5 to 10 years.
Yeah, I think, treatments that, number one, I think a big goal in this field is how to return people to homeostasis. So ideally, we can do something that you don't need to take treatments continuously for the rest of your life. I think that's one of our goals. I don't know if we really know how to get there right now. I think that the T cells that Dr. Paley mentioned are really going to be a key component to that. And I think also, moving to oral medicines is a big sort of movement in the field right now to make just treatment easier. I think very effective oral medicines that are comparable to injectables.
Great. Are there any rare symptoms or co-diagnosis that should be considered?
With
Dr. Paley,
is this for me? Oh man. So I think that in the umbrella of spondylitis, right, you want to have a very broad initial evaluation, just to from a clinician standpoint, to see what manifestations in that umbrella patients are developing, right? Because so if the patient has psoriasis, are they also having like joint pain? If they have both joint pain and psoriasis, are there, I have patients that have psoriasis, inflammatory arthritis, and inflammatory bowel disease. And so you have to be a little bit sensitive to those other manifestations to see if they're popping up. Not that this is not to scare patients that oh no, you have psoriasis and arthritis, you're going to get all these other things happen to you. But I think the clinician should be astute in terms of looking at those other manifestations. And then of course, all of our patients, many of our patients struggle with those chronic or conditions that go with chronic inflammatory arthritis and chronic pain and stuff like that. And the physician's always going to be instrumental in trying to tease out where are your symptoms coming from? Are they coming from active inflammation that we need to change your immunosuppression, or are they coming from sort of these new neural pathways that develop because somebody has chronic inflammation in their joint and has to deal with some chronic pain issues? And that's, a big challenge in the field that physicians, I think, are really struggling with, but we do a pretty decent job. Right now, we just need to do a better job.
Yeah, I think that was really well said. And I think it goes back to, I think, the first slide that Dr. Paley mentioned with all the different organs that can be affected. And I think it just requires a lower threshold to investigate if symptoms arise. And another condition that is higher risk in individuals with psoriasis is cardiovascular disease, which I think we're learning a lot more about, and being aware of that connection and trying to manage those risk factors.
what should patients expect in terms of long-term joint health with psoriatic arthritis? And are there any preventative measures to protect against joint damage?
So I think that, you know, when you pass through the door of the rheumatologist's office, the time between when you started symptoms, as Dr. Razani talked about, and then when you get to the door, that's a big determining factor in terms of your long-term joint health. But once you're there, you know, if you get there quickly, I think we have a lot of drugs and therapies now that really show that we can prevent a lot of long-term joint damage, and that's super promising. We obviously don't have anything that reverses some of the damage that's been established, and that's, I mean, it's still an unmet need. But I think honestly, the therapies that we have, finding the right one for you that works, that you tolerate, from my perspective as a physician, is probably going to be one of your biggest bets in terms of success.
I don't have much to add about joints, so
you can weigh in. That's okay. What challenges are researchers currently facing in advancing our understanding of this disease?
Dr. Razani, you want to take this one first?
Sure. Yeah, I think a big question is, what is driving all of the inflammation? So what is a trigger? And then what is the persistent cell that's continuously causing problems? And I think, Dr. Paley's research is really fascinating, really implicating particular T cells that might be an issue. And then there's also that may be added on top of other non-T cell factors. So really understanding what's the driver, I think, is a big question, and that will always be that information that will be really helpful is really getting at the tissues where the inflammation is happening. And so really being able to obtain samples where that inflammation is happening in the joints will be very helpful. And then understanding all of the data that results out of new technologies to really hone in on the problem, because lots of things go up and down in these samples, but really understanding what's driving them. And I'll say, I think mouse models are really helpful in this way, because we can really control things.
Yeah, I want to second that. I agree with everything Dr. Razani said. I would just, maybe this is a personal thing for our research, but one of the challenges we face actually is not from in terms of like enrollment in human studies, is not from patients, right? Because you guys patients are very invested in figuring out like why they got this disease and what we can do about it. But it's actually trying to find the right healthy comparison group, right? Because to really understand what's going on in the disease, you need somebody who doesn't have the disease to compare it to patients who do have the disease. And so finding enough people that are otherwise healthy, like that are willing to participate, I think that has been our greatest challenge, is convincing people to help us understand rare diseases that they have never been exposed to because they're rare. So that's one challenge that we think about a lot.
How can you best determine what biological treatment will work for psoriatic arthritis instead of just trying one after the other?
How good is your crystal ball? Yeah, I get this question a lot. I would love a blood test that would let you know, oh, this person should be on a TNF inhibitor, but that person should be on a JAK inhibitor, and this other person should be on an IL-17 inhibitor or IL-23 inhibitor, whatever. That would be fantastic. Like, physicians would love that. Patients would love that. We wouldn't have to like spend a bunch of time cycling through meds to find what works for you, as Lacey talked about in her introduction. I think people are trying to work on that. There are some attempts at this, but this is a big unmet need. We need a lot of research to, I think, start to figure it out, right? Because like maybe people have different genetics that are important for this, or different parts of the immune system that are activated for the disease that are part of this. But yeah, I don't have a good answer for you, because I want the same thing.
Yeah, I think the question is an excellent one. I don't think we have a really great uh, predictor right now that's super accurate. I think it will be really helpful to be able to study patients that particularly respond or don't respond to these medicines to understand perhaps what are the pathways that remain elevated in these different contexts. So I think there's still much to learn here.
That's a great segue, Dr. Razani, because the last question is, how can patients be a part of research?
Yeah, I think we're learning a lot from patient volunteers who were able to study their skin or joints before and after these very targeted treatments. And I think those types of studies really help because it's really an intervention where we know what the treatment is doing and how the immune system is changing. So I think those types of studies will be are being done and will continue to be very illuminating.
Yeah, so this is great. Yeah, I've had patients actually approach me asking if they could participate in the study, which has been fantastic to find, like proactive individuals. But I think if you're a patient at a tertiary medical center or something like that, where there's a lot of research going on, and you find the people that are doing research in your whatever your disease is in your domain, they can point you in the right direction and see if you can contribute to that endeavor, or you can look on clinicaltrials.gov to see if you're interested in that sort of thing, because there's a lot of clinical trials that are opening up that if that's something you're interested in, you could participate. But obviously, it's a free country, so you should not feel twisted to do anything that I say.
Well, thank you both so much for answering our questions.
Yeah, great. And as we wrap up, so just to kind of also tag team on that last response is that you can always check in with the ANRF website. We do have clinical trials that are open, and oftentimes we'll send out communications about new trials that you may qualify for. So always keep us as a resource. So I really wanted to say thank you to our fantastic moderator, Lacey. You did an incredible job. We so appreciate you being here and sharing your story. Also to our wonderful researchers for sharing their innovative work towards understanding psoriatic arthritis. You can see firsthand the impact ANRF has in supporting these incredible individuals and their research in a quest to find a cure. So I'd like to once again thank our partner, AbbVie, for supporting this webinar and our webinar series. And lastly, thank you to everybody who joined for this great event. If you've enjoyed this webinar, we kindly ask you to consider making a donation today by visiting curearthritis.org/donate. Every contribution helps, and it supports the incredible work that we do, from educational webinars to supporting our researchers in innovative research, so we can continue to advance and move towards hopefully a cure and understanding all of these diseases, including psoriatic arthritis. So we will also be sending out for all of our live attendees a survey after the event. Your input is so incredibly helpful to us in understanding how we can improve in these events moving forward, and the types of information and research you want to hear about in the coming years as we continue to do these our research spotlight series. I would also like to say, please don't forget to sign up for the next Research Spotlight series. So on October 2nd, we have scleroderma. On October 16th, we will have our lupus webinar, and then October 30th with osteoarthritis. So thank you all again for coming. Please stay connected to ANRF via social media. Make sure to drop your email addresses so that we can continue to stay in contact with you and share other important educational events that you may be interested in. We thank you so much for participating today, and we hope you had a wonderful experience.